about that, but I'm here to talk about a title which I made up, which was very, very long. But I wanted to look only at the beginning, the big melt. So what I did is I made a much shorter title for today. And it's basically From Greenland with Love. Right? So what I will talk about is one part of the work which we do. And I don't like this, but can I turn this on? Yeah, this is on. Okay. I like to move. Can you hear me in the back? Yeah. Maybe we should turn this off. So I started as a metamorphic petrologist. And what you'll see today, you will think, what the heck? But I started as a metamorphic petrologist because I was always interested in how, as a young person, I was a cave person. I used to go into caves and I saw minerals forming from water dripping from the ceiling. So I was always interested in these interfaces between minerals and waters and stuff like that. And then I started metamorphic petrology and I worked on serpentinites and ophiocarbonates. For some of you, something this means. This was long ago in the dark ages of my career. But then I realized that reactions, it's actually just about making and breaking bonds. But those reactions are between a mineral and a fluid, between a fluid and a mineral, between a mineral and a microbe, or what the microbe produces or eats. And slowly I moved toward the surface and realized that actually in the top part of the crust, nothing happens without organics, respectively stuff that the microbes produce and consume, whether that's CO2 gas, oxygen, they change redox, alkalinity, or whatever. So what I'll talk to you today, although my group does different things, is the part that we worked on in Greenland. Now, as a geologist, does this work? It should be. Yay. If you look at glaciation through the geologic time, right? We had, whether you look at long time ago, we had the Huronian, the cryogenic, we had lots of cold and hot houses. So it's all where it was very hot on the planet, where it was very cold on the planet. So we had this as the geological path in our 4 billion year history quite often. That's a normal geologic process. But when I want to literally make a big jump as a geologist from 2 billion years, 4 billion years back to the last, let's say, 1 billion year, because that's where we have ice, where we can actually date it. Now we have a new ice core that is a bit longer, but actually we have ice cores where we measured the concentration of CO2 and methane from the ice core. And in the last, let's say 800,000, let's say 1 million to keep it simple, we had values for CO2 in blue of 275 ppm. When we look at this 1 million year, like I said, now there's a new core out there where they will go even further. We realize that in this 800,000 years, it was relatively stable. Forecasts are not so good because forecasts for the present day, we are about here, and I'll show you that. Or if we go further, it's up here. Again, from the geologic history, this is not surprising. We have done it. It's the scale and the speed at which it happens, which is the surprising thing. So I now move back to the last deglaciation, which obviously gave you your Pannonian Basin here. Because in the last deglaciation, and this is the northern hemisphere, and the southern hemisphere, the melting. And you'll see here, it's actually melting a lot in the northern hemisphere, and also a lot here, but less a lot. So if you look at here, the number goes down to from 26,000 to 10,000, so it's repeating itself. And if you look at Greenland, Greenland is the sort of the remnant of what used to be a huge ice, including basically Europe was covered by ice. If you think about how much of this water that has melted, what did it do? So I'll transform this into a two-dimensional plot. And this two-dimensional plot is now equivalent sea level rise, or how much meters of sea level was there as a function of a year. And this is now in 1,000 years. And the delta time is about 8,000 years, where basically the North American melting that you've seen before, with a little bit of Antarctica, a little bit of Greenland, and a little bit of the Fennoscandian, actually contributed 130 meters of sea level rise. Again, now we're going 8,000 years. Normal, relatively fast, but relatively normal. This happened when the temperature change was 4 to 5 degrees. And what is done, it actually lost the ice primarily driven to the North American loss of about 3,000 gigatons per year. Now, that's not the number that we can put in context, but I'll show you what it means. So just remember 3,000, because if we now bring it back to sort of today, and I downloaded it this morning, this is from yesterday, from the Mauna Kea reading, we started at 275 for about 1 million years, and now, like yesterday, it was already 430. Right? Now, I'm old. I went to the Arctic for the last 3.5 decades, where many of you were not even born. But the changes that I've seen in one generation, and I consider it one generation, probably two, but anyhow, is actually dramatic. Glaciers have disappeared where there used to be glaciers. Islands appeared where there used to be no islands. Estuaries have disappeared. So huge amount of change. But why does this matter that it happens so fast? If you look at the 1850 to 2000, and this is every month. This is a beautiful heat map, what we call a heat map, and biologists love heat maps. Geologists not so much, but that's what we do. Where we actually look at how, from 1850 to about 2015, every month the temperature slowly but surely went up. Everywhere. This is global average temperature. And what you see that in 2015, in November, we surpassed the 1 degree global pre-industrial level. In very, very short time, as the geologist speaking, we did that. What does that do? Well, it adds water to the global sea level. This is a really nice video where people show how much water it added in 120 years. It's 20 to 25 centimeters. Now, 25 centimeters for Sopron is not a lot because you live at 270 meters. But try to be in Amsterdam, in Bangladesh, in New York, in Miami. 22 centimeters in 120 years is a lot. When you think about just Greenland now, Greenland at the moment is covered by 1.7 million square kilometers of ice, which is about 18 times Hungary. If the ice volume of 2.9 cubic million kilometers melts, it would add 7.5 meters to the global sea level. Again, for Sopron, not so much, but we'll come to that at the end. If you look at it just in 20 years, this is data from the gray satellites, because thank God we have satellites, which actually the last 30 years gave us a huge amount of beautiful data. What you see here is how, so just look at the change in color here from not much, so this delta changed, and how every year the amount changed. So what you see is actually more and more melting, and mass loss means how much ice has been disappearing just from the periphery of Greenland. And what you see here is a number of 279. Now let me remind you, I gave you a word of 3,000 in 8,000 years. This is 300 in a much shorter period of time. So this mass loss is accelerating. Just in this period of time, we have added 2.5 centimeters to the global sea level just from Greenland. We have reached a critical tipping point, so to speak, because the temperature in the Arctic is reached about 4 times global average. And we are about already 300 gigatons per year compared to in 20 years, compared to the 3,000 in 8,000 years. So the speed is very, very much. Now, how do we know? Well, satellites are smart things. They measure, and satellites, and we have a whole series of meteorological stations around Greenland which measure how many days the temperature is above zero, because that is called a melt day. And from 2012 to 2024, and even before, we have seen melting increasing. In 2012, before 2012, we used to have about 40, 50 days where on average the temperature was above. In 2012, it was 75 days. That means you see here 75 days. That means about 90 on a particular day in 2012, 98% of Greenland was melted. 98% of the surface was melted. It's not, it was an exceptional year, but it was not the only year. It happened in 2021, happened in 2024, it happened in 2019, 2017. So it happens again and again and again. So because we reached a peak point and it goes over. So we can measure things from satellite and we can want to predict what happens in 2003, 2300. Where do we go from here? The biggest problem that we have is that models can go in any kind of form or shape, because the unpredictable rate is, regardless of which IPCC international government panel you take, any kind of prediction you take, the albedo, which is the amount of light reflected, is the biggest uncertainty. And satellites literally just measure light down by a cap, and they measure it's a huge change. How does that look like? Well, if you reflect the sun for something which is green or blue, obviously that's a different albedo than if you reflect it from something which is white. Because people think, well, but Greenland is white, it should reflect everything back. That is correct. But this is a measured albedo change in the last 2 decades of the last century and the first 12 years of this century. So what you see, just look at them going from green towards blue. The albedo has been dramatically changed across all over Greenland. Now, why does this look so blue when it doesn't melt everywhere? It's because satellites measure things at the 500 by 500 meter pixel resolution, and it's an all average. But on average, you see that it moved from, let's say, what was above zero to below zero in a very, very short period of time. Why is that? Because people think snow and ice look like that. How many of you are skiers? How much snow do you have in Hungary, in Sopron in the winter? You wish it all looks white, right? We all wish that when snow falls in a city, it looks like that. It doesn't. Now, in Greenland, it doesn't look like that either. And this is one of my colleague scientists looking at the little helicopter flying away and leaving us there for 6 weeks. Because what we're trying to see is this is what people think Greenland looks like, but especially in the periphery, Greenland can look like this. This is not what we think is white, right? We have snow left on the crevasses, but actually it actually turns really, really dark. So why do you think ice is dark? What makes ice dark here?
Smog.
Smog, yeah. When it melts, water, right? So there are lots of things which make ice dark, right? Stuff that it contains in it. So the first thing I'll take you from the satellite image all the way to this very small scale. So this is what you see. You see this zone, this dark zone, which is the melting surface in Greenland from a satellite. And this is called the dark zone, which expanded since then. This picture was taken in 2017, I think, expanded in many other places, but we don't have another picture because the weather is not always so nice. But if I now take you from there, I'll take you here. Now it's 30 kilometers. So this is the bare ice surface. This is the snow line. And it's really, really dark here. And you also see these lakes and the rivers, so it's really lots of darkness. If you go now to 1 kilometer, you don't think that this is ice surface, but this is actually ice surface. Because it's actually even from satellite, you see the lakes, you see the rivers, and you see these bands of structure in the ice that actually bring up a lot of particles. If I bring it down to 50 meters, this is now rivers, little hummocks of snow and ice. I'll bring it down to 10 centimeters, and you can have a really clean ice, a little bit dirty ice, and super dirty ice. So even over a very, very small scale, you can have a huge variation. But a satellite measures something at 500 by 500 or 200 by 250 or 100 by 100 meter resolution. And that's okay because they have to average. But what makes it? Because darkness is not just one parameter. And obviously, like you guys say, when snow's here and you know what happens, when we go from fluffy white snow to wet snow, it goes darker. Just water content itself can change a big difference. When people think of albedo, they think of albedo as 1 means 100% reflection, and anything less than 1 means lower albedo. So when they calculated how much of the albedo that is in here could be coming from wet, that means snow physics, wetness, let's say from wet to dry, and I'm simplifying it here for a snow physicist, I'm sorry for the geophysicist, the physicist in the room. But the albedo is only explaining down to 0.75%, and that's insufficient to explain that. Now, the second one you mentioned is obviously black carbon or soot, both what we produce anthropogenically to our industries, to our cars, but also more and more so from wildfires. Every year in the Arctic, we have huge wildfires, whether it's Siberia or Canada. Even in Greenland, we had a lot of wildfire in Greenland, and you think like, where the hell, what is burning in Greenland? Lots of bushes in Greenland. In the moment the glacier retreats, there's lots of life growing. And it actually burned a lot. This is 2023, so I don't know why it's cut off. But these are fires that actually burned everywhere in the western part of Greenland. And they produce a different amount of soot that also goes on the glaciers. Even if you take what we call the black carbon, and you'll see BC from now on, and the water content, it still doesn't bring it down. It only brings down to 0.7. That means it's still insufficient to explain all the darkening seen in the satellite. And then obviously, it's minerals. We are geologists. We want to know what happens because you have more and more dust from the Sahara gets blown up, or dust from the retreating glaciers, because when a glacier retreats, it leaves glacial flow, really, really fine-grained material, which becomes an Arctic desert, gets brought up and gets put back on the glaciers. We had no idea what the albedo reduction is when we started this, due to the mineral dust. We had no idea whether the mineral dust or the black carbon or the snow physics produces more and not. Because water is a parameter that is the same regardless where it comes from. Black carbon, let's assume it's not the same, but let's assume it's a soot kind of thing. But minerals, depending where they come from, and they change particle properties because of particle size, they change composition if you have sedimentary rocks or radiometamorphic rocks, they change darkness depending on composition and particle size. So we had no clue. And then the last one is because it's not just water, black carbon, or mineral dust, but it's also something that most of you don't think exists on glacier and ice. It's actually biology. Now, for those of you who have been on snow and ice, sometimes you see the snow turn red. And that is not because, it's because of algae, which actually grow only there. It's called sometimes watermelon snow, but I'm not sure if that you have heard of that. And it's actually can turn the whole thing really red. And it's these little single cell algae that are about 20, I don't know what scale bodies it is, about 15 to 25 microns in dimension. And they are pigmented red. And on the ice itself, you have purple algae that are different kind of family. These are streptophytes and these are chlorophytes for the biologists in the room. They are different lineage, but actually these ones grow only on ice. And these ones are only happy on snow, but they actually also survive on ice. Now, when you go out in the summer and it's hot and you want to protect yourself against the sun, what do? You put a hat on or you put sunscreen. They're so much smarter, they make their own. So what they do, the pigmentation that they produce is actually to protect their chlorophyll. Because they have such a high UV exposure on the ice, they need to protect themselves. So they make dark pigments to protect their chlorophyll. But by doing that, they melt their environment. Because obviously a black or a dark particle on a white surface melts more than a white, because you have a white t-shirt or a black t-shirt in the summer, you wear white, right? It's literally as simple as that. They have to protect themselves while they actually thrive and bloom. And we see really big areas of bloom like that. And they actually darken the color in addition. However, when we started with this, I don't know why I can't see, what does it say there? There we go. So at the beginning, the biology is really fast changing because it has to have liquid melt water. So it actually has to start in the summer, in the short period of melting, it has to go. But they were not included in any global models because we didn't know what it is. We knew what water content could be. We knew what black carbon content could be in the global models. We now try to put in the minerals, but also we tried to put in the biology because we needed to know. We didn't even know it was there. How they tick, what do they eat? What do they like? What do they not like? Are they alone? Are they a bunch of people? Are they friends? Are they happy? Can the dust that is deliver enough food for them? Or is the melting of the snow and ice enough food for them? What do they do? We had no clue. And that's when we started these questions. We started this is a long time ago that I started this. because we need to combine glaciologist, geology, remote sensing, microbiology, geochemistry, mineralogical, blah, 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 blah. Because we have to ask a bunch of questions that cannot be answered by a single group alone. First, what comes from the air? It's coming snow, soot, nutrients, everything can come from the air because global circulation delivers a lot of things to Greenland. What happens when it starts melting? What triggers the melt? Do microbes and minerals play nice? Do they like each other? Do they hate each other? Do they need each other? But how and what do they do? And does it have consequences for albedo and melt? And then, this is all the summer story. We thought, well, what happens then in the winter? This is where, do microbes sleep at night? But you have to remember, in the summer, there is no night in Greenland because it's 24-hour sunshine. But especially in the winter, when they freeze in, what happens to them? What do they do in the winter? I always say they're really lazy. They have a party for 3 months in the summer when it's melting, and then they go to sleep for 9 months. But is the sleep really sleep, or is it dormancy, or are they just dead? We didn't know. And then we started this, and this is an example where we started with something called black and bloom, because we thought it's soot and microbes. Then soot that is delivered, black, and then bloom, which is the microbes. And we started putting our tents in different places in Greenland. We started up here, and then we went around. And this summer, we actually do these 3 last red dots that we do to see whether all of that happens in different sites in Greenland, in different years in Greenland, and with all these different measurements to cross-correlate these. So we started in 2012 with the Black and Bloom project, and now we continue with the ERC project, which is called Deep Purple, because the algae that are dominating the ice are deep purple. And deep purple sounds kind of cool, because it's also memorable. We even went to a Deep Purple concert just because we run the Deep Purple ERC signature. We actually booked those tickets just before, when we got the grant in 2020. We were supposed to go in October 2020, but then COVID happened. So we had to rebook the ticket in 2022. We went to Copenhagen to a Deep Purple concert, which was, none of us are Deep Purple aficionados, but it was okay. So what do we do? The first thing is, my question was, what comes from the air? Now, I don't know how many of you are aerosol people, and if none, it doesn't matter. I was not an aerosol person. So we designed ways to collect based on solar panels, because you don't have a plug to put your power in. We collected all kinds of, air things. So we collected stuff on TM grids, we collect on silicon nitride windows, we collect them on filters that we can put into different tools that we can measure the black carbon contribution, the mineral composition, the microbial composition, so anything that comes from the air, to try to see whether we can quantify at the place where we were for 2 years in a row, whether we can quantify what is there. And we did. So in the black carbon part, let's call it black carbon. I put it in quotes because black carbon is a huge variety of carbon components that go from biomass burning all the way to fossil fuel, but I don't have time to go into that. We actually characterized both the composition, the size distribution, the fractal dimensions, the delivery rates in particular 2 years. And we see that they're all the classic black carbon, let me call it black carbon in quotes, onion rings fractal dimension, which is actually what actually makes it really black. But the amount of it is relatively small. And we actually measured with spectroscopic means to measure what is called the sp2 hybridization. And it doesn't matter what it is, it actually gives you an idea of whether this is black carbon or brown carbon. It means it's a fossil fuel burning or biomass burning. And this is a very big simplification of this, the differences. But we actually measure this with both with synchrotron spectroscopy, soft X-ray spectroscopy, and with scanning electron microscopy and TM. Because if we know how much of the sp2 hybridization comes from what, we can derive the light adsorption properties of the black carbon in our samples. And that means we can know how much this affects the albedo. And this is just what comes from the air. What have we found? That actually 1% of the elemental carbon, which is considered the black carbon, in air and in fresh snow, and we had to collect 100 kilograms of fresh fluffy snow to get enough material to do all these analyses, which because we got 30 milligrams of dust and black carbon and microbes from 100 kilograms of snow. So we had a person who did nothing else for 3 weeks but just melted and filtered snow. Poor them, but they weren't nice. Right? But between 1 and maximum 6%, we could see that in air, snow, and in fresh snow and ice. That means it's actually only a relatively small contribution to albedo reduction if you compare it to what happens in the weathered snow, and specifically in areas where you have a lot of microbes. Because there, 94%, up to 99 to 94% is actually organic carbon, which is microbes and non-parogenic carbon, which is not the black carbon. So obviously we measure particle size distribution with ethylometer, which are devices that you stick in the air and let it measure. We measured mineralogy because we want to find what comes down. We found calcium phosphate, which is what feeds part of the algae because they need phosphorus as an important thing. And we measured all these because we knew that anything that comes from the air contains material that the microbes can actually use. But if we look at the difference between contribution from mineral dust versus black carbon, we have to remember that mineral dust is relatively large, while the black carbon is just very, very tiny. But the black carbon is really, really, really black. Well, if you grind samples smaller and smaller, they actually become more and more white. That was always the balance between the two. And when we included the amount of black carbon and the amount of minerals that we have and the composition of minerals that we had at this particular place, we saw there was a small proportion of black carbon. As geochemists, we also measured where does this mineral dust come from? Because it has to be transported from somewhere. So we measured, as we do geochemists, we measured rare element composition, we measured isotopic composition, and we measured the signature. Most of our samples came from local sources from Greenland. There's always a proportion that came from Asia, a little bit from Africa, but a huge proportion in our samples at the time in 2016, 2017 came from literally from here, put under the surface. So it's literally stuff that comes from here, gets put on the surface, and then it gets melted out. Now, there's obviously, I showed you those little bands because that's melting out old Holocene dust. We can't distinguish between whether it's old dust or new dust because it's all the same rocks. So at the moment, we're trying to figure out how to make that differentiation, but it's mostly low. And then we want to figure out how much is getting on there. And this is a paper that is now just came out this year. But we see that basically we have a very, very small particle. The particle size is always really small. So literally like 2 to 3 micron average what is transported. There are some bigger particles, but by volume, they are huge particles, but maybe 2 out of 10. But 80%, 90% is really small particles in the below 2, below 1 micron composition. That means we quantified all these. But actually important thing is that, again, it's dust that forms from here, which gets up there, and the algae love it. How do we know that they love it? Well, if you think about melting snow, this is a picture where, if you look at it, you have no clue actually what it is, but these are humans for scale. And this is a melting zone between relatively clean snow and super dirty ice. So if I'm now showing you, now I'm going into the biological part, but I keep it simple. So if I'm showing you what algal blooms happen on snow, and it's classic photosynthetic algae. What do? They make chlorophyll. They go green, they form chlorophyll, and then they go red. And these are not ice cream, not pistachio and strawberry ice cream. This is red and green algae from a glacier. And that's the way it looks like, one example. And that means these surfaces are huge and they can actually lower the albedo dramatically. Now, when we analyze them, obviously we did sequencing. And for those who have not done sequencing, the only thing you need to look at, and I don't know why it cuts off the left and right, but actually you need to see that we measured, used the 16S and 18S amplicon sequencing to actually see who's there. And what we see is that most of the colors here are purple, which is very few. It's a small diversity. Now, if you take a spoon of seawater or a spoon of Lake Balaton or a spoon of the river which goes here, and you have a diversity of like 80 or 90, here we have a diversity of 6. There are very, very few algae that are specific, only living on snow. And when we measured 21 different parameters, the only thing they needed is water and sun. We couldn't see a big correlation. There's independent of location. We measured 126 locations across the Arctic. And most of it was actually they only need water and sun because there is enough nutrients coming either from the minerals or from the melting snow, or they suck it up from the atmosphere. They don't need anything particular. But then when we went to the ice part, obviously there are different microbes there. This is where we go very dirty, because we can go from super clean to super dirty. And these little particles, see, this is 50 by 50 microns. And these little particles there are the ones which harbor these basically streptophytes, which are the plants. These are actually plants. And they form this pigment, which is called purpurgalin, which is like what makes your tea dark. While the red algae that you see on the snow is carotenoids, is what makes your salmon and your carrots red or, yeah, red. So it's just a different kind of pigment that they produce to protect the little chlorophyll as they are produced by have too much UV. When we looked at those in terms of the sequencing, there's even less diversity. We found 1 or 2 species dominate everything. Now, that is really special because in most aqueous environments on Earth, diversity is what drives interactions. But these are such specialists that they actually just live there, and they are the ones which dominate the whole composition of the microbial community. But that means they also dominate the carbon, the nitrogen, and the phosphorus cycling, which is actually driving the CNP ratios, which every living organism needs. So this is an example of one place. We've done this everywhere in Greenland. Now we'll get samples from there. And actually, we evaluated this also from a variety of different databases that exists. And the diversity is very, very similar kind of everywhere. And most people thought, well, we only have the dark zone here, but we went here, we went here, we went here, we went here, we went here. This year we go here again and again in 2 different places. We find them everywhere. So the darkening that is seen from the satellites at the big scale with 500 meters pixels is actually at the small scale kind of everywhere. And we try to figure out what does that mean in terms of the albedo. We've done this globally as well. We compared all this, the details here doesn't matter. We looked at the diversity globally to try to see if the global distribution of these algae is actually changing from Antarctica to the Himalayas, even Kilimanjaro, or to the Rockies. And obviously we have Greenland and we have Svalbard. And actually it's very, very similar everywhere. And they are dominating, very, very small species are dominating the whole process. So my third question is, do microbes and minerals play nice? But what I mean by that is, do they need each other or do they not care? They just happen to be at the same place. So what do the microbes do to make them bloom? Do they get something from the minerals and from the ice? Because as you guys know, solubility at 0 degrees, and most of the time snow and ice at 0 degrees, is not very high for most minerals. Because as they thrive and they bloom, they change the carbon, phosphorus, and nitrogen cycles. And obviously, how do they lose albedo? Now, when you look at putting a mineral into a bucket of water and you look at solubility at 0 degrees, it's very low. But then all of a sudden, if you put a bunch of organics in there that are produced by microbes as enzymatic processes, they may actually do it faster. That means if they need to take something out of your water or out of your mineral, they will. So what we've done is we actually image them, and it's now huge scale, where you see these are minerals and microbes all together at this scale, and they actually live on the minerals, not surprising because it's a nice substrate. And if I go now, give them food, what you see here is a measure of their activity. This is a measure of relative electron transport as a function of light, how much light they have. And the only thing you need to take here, we incubated these ones with nitrogen, ammonia, and phosphorus. And we've seen that when you look at the phosphorus alone, which is in purple, it actually dominates. In the moment you give them phosphorus, it's like Christmas. They love it. because they need phosphorus. Now, they also need ammonia, they also need nitrate, but that's not as a limiting factor at the time. So when we did this and we published this in 2021, we thought, okay, well, but where do they get the phosphorus from? Because at the moment, what we did here, we just gave them dissolved phosphorus because it was easy. And then we said, well, okay, they actually have to take it also for minerals because they live on minerals. This is our 3-micron scale bar. And you see these are the microbes and they all live really tightly on minerals. That means they solubilize how much and whatever they need. And what we then quantified, as a mineralogist, you put it in X-ray diffractometer, you measure how much calcium phosphate is there because there are not very other minerals where you have a lot of phosphate. So we actually quantified the hydroxyapatite, which is really this little teeny, teeny yellow line there. So it's actually very, very little. But when you quantify it based on the number of cells per unit area, it's plenty. Because when we plot total organic carbon, which is the equivalent of how much biomass is on an area, with how much phosphorus is in the sample, it's a one-to-one relationship. That means there is enough for them to take out. That means somehow they found a way to solubilize more phosphorus if the minerals are there. Then we used a whole series of different omics. We used sequencing. Because normally when you do sequencing, like we know about, during COVID, we all did PCR to see what it is. But if you're sequencing, the first thing it gives you with genomes, it tells you who's there. It doesn't tell you what they do. It means we do a variety of measurements. We do measure protein concentration, for example, to see what could they produce. We do metatranscriptomic to see what is their activity. Are they doing a particular activity? But here I'm just showing you that basically if we just do the amplicon sequence, the who's there, oops, I don't know why I'm pressing the wrong button, sorry. We see that in the snow and in the ice. In the red one, you have obviously most of the red algae. In the purple one, you have mostly purple algae. Again, not surprised, this is what we were expecting. But then we measured the biovolume with a different method and we see the same thing. And then we measured the proteins and we see again, the red algae have most of these proteins and the purple algae have most of these proteins. And I'll come to the proteins themselves. Why is it important to know about proteins? It's because that's a factory that produces eventually their DNA. And again, forget about the details for those of you who are not biologists, but actually there are particular pathways that actually makes them tick. It's something called shikimate pathway, and I brought it up because it's a nice name. It's not the Japanese name, but actually it produces particular proteins that actually help them survive and actually lead to pigment production. Because in order for them to produce pigments, they have to require the pigments and they have to have the pathways to produce the pigments. So we didn't even know what is the pathway. So now we know that it's a high abundance of these enzymes that actually allows them to produce pigments. Now, it also happens that these enzymes are very good at solubilizing minerals. We don't yet know how that connection works, but we are on the way to it. And then we ask ourselves, well, okay, now we know something about proteins, we know something about minerals, we know something about carbon, we know something about everything, but how does this affect albedo? Because they darken the surface. And here I'm just showing you a couple of examples because how do you measure albedo on the ground? Because satellites measure 500 by 500, 100 by 100, 200 by 100. But I mean, you see the microbes, they're like 25 microns. So you need to figure out how to go from there to there. Well, you have a bunch of people who like to play with drones. Boys and their toys, but girls and their toys too. And so basically we measured spectral measurements on the ground, literally at 50 centimeters. We used drones to measure at 10 meters, 100 meters, 250 meters. Then we actually worked with the satellite people to actually look at MODIS or Nmap, two different satellites that actually measure albedo, to actually link these things together. And what do we get? Because obviously, when you look at the ground, the green snow, remember albedo is 1? And we said that water and black carbon bring it down to 0.7. We didn't know about minerals, but now with the microbes, with the green snow itself, if it's green, it brings it down to 0.4. The red snow brings the also down to 0.4 and 0.45. The reason why the green snow is lower is because the green snow is normally wetter. It needs more wetness because the microbes use the water as a film to protect themselves against the UV, And the red snow algae, they live on the top of the dry snow, so it's a bit different. But when we looked at the effect, oops, it is 13% more melting just by the fact that we have the algae. 13% compared to the others. So it's almost like a runaway effect. They live, they are green, they produce the pigment, they melt their house. By melting their house, they make more algae because they have more water, they lower the albedo, and it's like it's a perpetual thing. It's a positive feedback mechanism, but now we have an idea of how much just the red algae do. When we look at them at the purple algae now, we actually did a bunch of measurements on many of the bare ice surfaces, and we see, using a variety of models, very, very smart people, I don't understand anything about these models, but they actually looked at the measurement from 25 centimeters square, 50 centimeters square, 100 meters square to 500 meters square to match what we see in satellites. And they've seen that the glacier ice algae, which are these purple algae, they can produce 13 to 15 more melting, and in areas up to 26%. That's a quarter of more melting than the ice alone with water and black carbon. That's a huge proportion of more melting just due to the algae. Obviously, we try to look at this back in time, and what you see here is the processing and accelerating from 2000 calculated back on satellite data to 2016, how the area in the western part of Greenland is actually changed, and the red part is more and more algae-covered bare ice. So you go from 700 kilometers to 10,000 kilometers, and in a very, very short time. So this is just one area where the satellite has enough data, but now we see here, we went up there north, and this year we go up there. So it's actually happened everywhere on the Greenland Ice Sheet. And that means this kind of up to between 30 and 26% melting will just accelerate and it'll go much faster with time. So it's a runaway effect. How am I doing? Fine. So we have algae, we have minerals, we have black carbon, we have water. They all, in their own way, they reduce the albedo and they enhance melting. But it's like a vicious cycle, because the more you retreat the glaciers, the more dust you can put back on it. The more you melt the glaciers, the more the algae say, Christmas, they can bloom. So it's literally, you deliver some dust, you deliver some black carbon, you deliver nutrients. The algae grow, they reduce the albedo, they increase the melting, they produce more liquid water, and it's going around circles. And that means all of that leads to sea level rise. And I told you at the beginning that, sea level rise has reached, just from Greenland alone, in the last decade and a half, 2.5 centimeters, which is a lot. So what happens? My last question, this will be short. So let's assume the weather, 3 months of sunshine, 3 months of party is over. Winter's coming, right? We play Game of Thrones here. Winter is coming. What happens with them? So normally, when you think about the environment here, when it gets cold, everything freezes and either dies or stops. We have the advantage, we put coats on, they don't have that. But we were asking ourselves, what happens with them? So we actually went in the winter, which is non-trivial because obviously you can't see much, and try to understand what happens in the polar darkness. Because if you think about the Arctic, they have 4 months of darkness. And so photosynthetic organisms don't like no light. We are not very happy if we are in the dark for very often. We see that what happens to our cousins in the north. But we also want to know what wakes them up in spring. Is it literally just water and light? That's what we assume, but we need to figure out whether they can prove it. What food do they need? Food in quotes, because it could be just sunlight before they think, I can go again in spring. We don't know. So just to put it in context, this is in September, we have 13 hours of sunlight. This is just the sunlight, the challenge, because this is the amount of sunlight that they have in September, okay? 13 hours. In January, that's 0. It's 4 months of darkness. So these 4 months of darkness stops everything because they're all frozen. This is the way it looks like in the winter. You just have moonlight, and it's very bright because obviously you have reflection from the snow. but there is no sun. We went there the week before the sun actually arrived somewhere at the horizon, twice in 2023 and 2025, to actually collect samples below the snow to see what happens. And what you do, you have to do everything in the dark. Because you go in the dark, you collect with sun, you actually in the lab, you have to process everything with dark light, because in the moment, what do we do? When I wake up in the morning and the light is coming up, I'm like that, but I don't want to disturb the little microbes. I want to see what they do when they're in the dark. So we have to process everything in the dark to make sure that they know we can actually measure their activity, their metabolism, their transcriptomics, and everything while they are doing in the dark, but also measure all the geochemistry. So what we see is we have, and this is a difficult picture, but this is the last summer ice surface. This is the snow up here, and this is the deep core. So we actually measured all this. The reason why it's red, because we use the red light to measure everything. We didn't want to. But we measured this core and gave it a little bit of light, like 20 minutes of light and 1 hour of light, to see whether the microbes in this layer, do they wake up? And so what we've seen, we've seen that the microbes in that layer are still pigmented. They haven't lost their pigments, because the tannin, the purpurocalin-like pigment, is not yummy. There's nothing nobody wants it, because tannin is not a good molecule. But what we look at it, we've seen that with short light, which is 20 minutes, and long light, which is 1 hour, their function, and I'm just looking at this is a specific photophysiology measure, actually increases. There are 3 different cores that we took, increases dramatically from 0.2 to 0.4. That means even with a little bit of light, they instantly think, ooh, and they're still frozen. Remember, this is still in the frozen core. So what we then did is we brought these cores back in the dark, and now we actually melted them back in the lab to actually analyze them to see whether they do the same in the lab if we give them more light, if we give them melting more food, etc., etc. But that's an ongoing process. But they are very, very quickly, in the moment they see light, they actually go. Because even if you have 1 meter of snow cover, there's enough light penetration to the snow to actually reach the surface, because snow is very porous and light goes through it. So it's actually very fast. So we looked at these cores. This is the last year cover. This is the snow. This is a layer, but this is now a core that is not processed in the dark, so I can show you. But you have all these different layers, all these different mineral layers and microbes in there, and we actually analyze those to see what they are. Again, bringing it to to proteins. It's something like a phytohormone. Doesn't matter what phytohormone means, but it's actually what it does. It converts sucrose to fructose and glucose, because this is the winter. They need that sugar, and they survive of that sugar that they have in them. And that means these are the dormancy associated proteins. They go sleep, but they have a little bit of activity, and they produce a little bit of sugars. They transform the sugars, because by the transformation of the sugars inside them, they actually can survive while they sleep for 3 months. And that means it helps them get through the winter. And if we then know that with light and with a little bit of sugar change, they actually can go, we know what triggers them. Now, there's much more other measurement going on. We do a lot of organic geochemistry analysis, because we want to understand what do they produce, what do they consume in terms of organic molecules with high resolution mass spectrometry. We look at the pigments in many different ways with looking at the metabolic pathways. And there's a whole bunch of people that are still ongoing work, from Pamela to Miriam. All of them love to go on the ice. Nobody likes to be in the lab, but we all have to be in the lab, because bring expensive samples back, you have to analyze them. And that means this is we got a lot of answers in the last sort of 10 years, but we obviously, like every science, you got a lot of more questions. So much more ongoing with Deep Purple. But the reason why I'm bringing this is because actually today is the future. I did this calculation in 2021 in August. In a single day, we lost 8.5 billion tons of ice from Greenland. Sopron is 170. Oh, no. It's 329. Sorry. So let's assume Sopron is actually 291, and this is the wrong number. It's an old number. So Sopron surface area of the city is, I think, 290 square kilometers. Let's assume, despite the fact that you said it's growing very fast. That's about, that one day of ice would have covered 50, I think the number is actually different, 35 for Sopron because it's bigger, 35 meters of ice over Sopron. What's the highest building in Sopron? A church? Fire tower, right? Probably, how high is it? 35 meters? Yeah, so you probably see the tip of it, but everything else would be covered in ice. That's a huge amount of ice in a single day, just in 2021. It happens again and again and again. Now, there's this paper from 2025 where they show that this is now the annual disappearing glaciers worldwide and predicting to 2020, 2100, depending on which scenario you take. Now, remember in 2015, we passed one degree. We passed 1.5 degree globally, especially in the Arctic long ago. So this is now predicting to get to the 4 degrees. Remember I said in the last deglaciation, it was 4 to 5 degrees that actually brought 130 meters of sea level rise in very short 8,000 years. So if we predict that, we see that actually it could go very, very fast. This is a global measurement for 200,000 glaciers. And this is just Greenland periphery. That means only the glaciers which come off the Greenland, which is 20,000 itself. And the predictions are not very good because the cumulative disappearance of glaciers globally, it doesn't matter whether you take this scenario or this scenario, it doesn't matter if 20% of them or 80% of this disappear, they will disappear very fast. 2100 is 70 years from now. That's 2 times the time I was in Greenland, in Iceland, in Svalbard, in the Arctic. It's not a very long time. It's 2 generations of students, postdocs, and scientists. So it's very, very short. And this is not something that happened on the Greenland Ice Sheet because the exceeding this 1.5, which happened in 2022, actually affects whether we're talking about coral reefs, we're talking about Amazon rainforest, lots of different things. But I just brought the story about Greenland because that's what we've been working on. And depending which parameters, warning threshold you take from the predictions, they all will eventually collapse. Now, if we got 2 degrees, obviously Greenland will be the first one to collapse because we already reached the peaking point. But the West Antarctic ice sheet will collapse, boreal permafrost will collapse. We talked about that. We release more CO2 or methane. The subpolar gyre will collapse. That means the ocean atmospheric circulation will change, etc., etc. And this is the temperature that in Hungary. I couldn't find it for Sopron alone, but this is for Hungary. And this is since 1980. So when you see, obviously, the temperature has increased by more than 2 degrees. So you are very close to the 2 degree. Obviously, it's not a global average, just in Hungary. And if you look at that, this is the map now, predicted for all the ice in Greenland would melt. You know where Sopron is? I put it up for you. You would be fine because you are 270 meters above sea level. Berlin, where I live, would not be fine because we're only 34 meters above sea level and we would be swimming. Now, this doesn't happen in 10 years, it doesn't happen in 20 years, but this will happen in about 100, 200 years, and it means we have to prepare for that. And I'll quit with this because part of the reason why I do this is because as scientists, I want to understand the process, but also because I feel we don't do enough for the next generation, for my children, children's children, and so on, to actually how to have a livable planet. And this is one of my favorite cartoons because obviously there are lots of people who think climate change, hoax, and stuff like that. It's not something that is an EAG view, but it's my view because it's not a hoax, it's real. But I also want to finish off thanking all these people who have done the work because I just pay the bills. I love to go to Greenland. I love to go to the field work because that's when I can do science. But actually, we have a lot of postdocs, PhD students, and collaborators, and obviously people who gave us the money to actually do this. I want to finish. Sweet. That's what you
Dream, a laugh, a kiss, a cry, a rise, a wrongs, a moment.
Thank you very much.